Welcome back to the channel. So in this video, I'm going to give you an overview of molecular cloning, and we'll also talk a little bit about CRISPR technology and how that is used, or how the molecular cloning process is used to contribute to that as well. So when we talk about molecular cloning, typically what we're talking about is the creation of artificial genes. Quite often we will want to create artificial genes that allow us to either track the expression of a gene. It could be to create proteins that have been fluorescently labeled or tagged with some sort of indicator so we can see when it's expressed or where it's expressed. Sometimes we're introducing novel genes, so new genes into a species. Other times we're introducing mutated versions of a gene to see what happens if we mutate a gene. But regardless of what your application is going to be, the creation of artificial genes through traditional cloning techniques typically relies on the use of bacterial plasmids. So artificial genes typically get created and then inserted into these circular chromosomes called plasmids. And then they typically get transformed into E. coli, a type of bacteria. And then we use those E. coli as sort of gene factories to amplify and create billions and trillions of copies of our artificial genes. And then we can choose to put those into cells or other species that we wish to study them in. So cloning plasmids have been around for a very long time. And over the last several decades, giant libraries of these bacterial plasmids have been created. They all have a lot of things in common. So, for example, most of them are going to have a large number of restriction enzyme cutting sites. So restriction enzymes are these enzymes that are produced by a species of bacteria. They cut DNA at specific sites, so a specific sequence. So by creating plasmids or vectors that have all of these different sites, you can see them here in italics. These are different restriction enzyme sites, and you can cut open the plasmid by using these restriction enzymes, and those create sites where you can insert artificial genes. So having large numbers of restriction sites, because they all have their own unique sequences that they cut, gives a lot of flexibility. So for example, if you're going to insert an artificial gene into a plasmid, but that particular transgene, that artificial gene you made, has, for example, an EcoR5 site in it, well, then you can't use that to clone into here, because not only will it cut the plasmid, it will also cut your DNA at the wrong place. So you might have to use a different one. So we create these plasmids with lots of different options. So we have lots of different enzymes we can use to insert these. They will also contain one or more antibiotic resistant cassettes. This allows us to identify the bacteria that are carrying these plasmids. This is important for isolating transformed bacteria and then using those guys to isolate the plasmid back out. The other thing is plasmids must be fully sequenced, because that allows us to know which restriction sites we can use and also allow us to know which genes we can put into them. So we know with these plasmids, this is where we can insert artificial genes so we can express them in E. coli and then harvest that DNA for use in other species. How do we create artificial genes? Well, in large part, this is done through a different type of process known as polymerase chain reaction, which is usually abbreviated to PCR for short. So polymerase chain reaction is a synthetic way of replicating DNA. Essentially, within a tube, we create an environment very similar to that of a cell, and then we add enzymes and buffers and salts to amplify a specific sequence of DNA. I don't want to go too far down the PCR rabbit hole. There are lots of other resources out there to learn more about it. But essentially, what we can do with PCR is, as long as we know the sequence of a gene within an organism that we wish to copy, we can do that using PCR. Essentially, we replace things like helicase and topoisomerase with heat. We create synthetic DNA primers to designate spots within that template DNA where we want the amplification to occur. So essentially, you put one at one end of the sequence you want to create more of, one at the other, and then allows the double-stranded DNA to be created this way. We use synthetic buffers and magnesium chloride to simulate the cytoplasm, and then we use a special type of DNA polymerase isolated from the bacterium Thermus aquaticus, with the main reason being is this particular DNA polymerase is stable at temperatures reaching the boiling point of water, which is important because you can see that in order to do what we need to do, we need to operate in temperatures that are significantly higher than those we would find in most living things. Okay. So again, if you're more interested in DNA, if you want to know more about PCR, you can go back and look at that from other sources. But essentially, what you need to understand is when it comes to molecular cloning, typically we are going to create our artificial gene sequences using this PCR procedure. It looks a bit like this, and you can see how after each round, we go from 1 copy to 2, and then from 2 copies to 4. And by the time you've done 30 to 35 cycles of this, you've created trillions of copies of your original template DNA that you want. Typically, once you've done a PCR, you're going to want to investigate your results using gel electrophoresis. So to do this, we create gels made out of agarose. These gels are then loaded with DNA, and then an electric charge is passed through that gel, where the negatively charged DNA is pushed towards the positively charged cathode. So this would be set up so that the positive terminal is down here, the negative terminal is down here. We load the DNA into these wells by combining it with something called a loading buffer. And then this DNA is pushed through this gel matrix. And then the result is the formation of tidy little DNA bands. And the reason why is these bands represent DNA fragments that are approximately the same size. And as you can imagine, with a constant force, smaller chunks of DNA will migrate farther down the gel than larger ones. And then we can use these ladders for comparison's sake. And often we'll get a gel that looks like this. Okay, this will confirm that, hey, we thought our original gene was supposed to be 1,022 base pairs long. Well, there it is. Okay, that confirms that our PCR worked. The reason we can see these light up is because within this gel, or alternatively, you can soak it after running the gel, we include chemicals such as ethidium bromide or cybersafe, and this causes the DNA to fluoresce under UV light, okay? And that's what you're looking at here. So let's say that we've created a transgene. We've created a transgene, and now we want to clone it. We want to put it into one of these bacterial plasmids, and we want to do something with it. We want to find something out. Well, one of the first things you have to ask yourself is, what is it that you're trying to learn? What are you trying to discover? So we'll talk about 2 different types of fusions, gene fusions here. So quite often what you want to do with a transgene is use that to figure out when a particular gene is expressed and where it is expressed. Okay. If you are interested in simply knowing when a gene is activated and in what cells it's activated, you can create something called a transcriptional fusion. So the way we do this is simple. All genes have an upstream sequence called a promoter. And as hopefully you remember from some of my other videos, promoters are where RNA polymerase is going to land prior to transcription. Okay. So if you want to know when a gene is being expressed, what you do is you amplify the gene promoter via PCR. You clone it out. Okay. You then take that promoter and you put that upstream of an artificial fluorescent reporter within your plasmid. What happens then is when you express that particular plasmid in your cell or organism of interest, whenever that gene promoter is activated within the cell, this plasmid will also be activated. That gene promoter will become active, but instead of producing the protein instead of transcribing the gene that would lead to the production of the protein, it'll drive expression of this fluorescent reporter, for example, GFP. And in any cell, at any time when this gene promoter is activated, that cell will light up fluorescent green or blue, depending on whichever colored fluorescent protein you're using. What's really neat about this is now you can look, and in many cases, you can do this in vivo in a living organism and say, hey, you know, we didn't see any fluorescence, and now we expose this particular cell or this animal to this particular treatment, and now that gene gets turned on. And that gives you a very good idea of, oh, well, stressing it with heat, feeding it, doing whatever it is, causes the expression of this particular gene in these cells. So when we talk, so for example, here's a way we would typically do this. First, we would identify the promoter of region of interest. We say, oh, okay, we're interested in knowing when gene A is expressed. Let's go grab about a 4 kilobase chunk upstream of that gene. Could be bigger depending on the organism. So we amplify that promoter. We then clone it into a plasmid using restriction enzymes. Immediately upstream of that, or immediately downstream of that in the promoter or in the plasmid, we put that particular GFP reporter. Then, when we express this in our organism or cells of interest, after allowing bacteria to make trillions of copies of this, we purify it, put it into our cells or organism of interest, and then we watch. We use fluorescence microscopy to figure out how that would work. So we identify the promoter region of interest. We examine the plasmid sequence and we choose 2 appropriate restriction enzymes. So how would that work? Well, simple. First off, we have to make sure that the plasmid we're using has the restriction enzymes that we're interested in. We also need to make sure that those restriction enzymes are also not found within the promoter sequence, because we're going to have to cut both ends of that target sequence of that promoter with those restriction enzymes. We're also going to have to cut the plasmid with that. And then what we can do is we can then anneal that promoter in here using ligase. It's a process called ligation. So you can see, for example, with directional cutters, you can see that it cuts after the G in that sequence. This creates what we call sticky overhangs, because if we take a artificial gene that has the complementary overhangs, they'll sit just nicely in here. So for example, here's our PCR product. We cut the ends with this particular restriction enzyme, and you can see that we have a little bit of single-stranded overhang here, a little bit of single-handed overhang here. We then cut the plasmid with the same restriction enzyme, and you can see that it creates complementary overhangs. You can see that this AATT is going to want to fuse with the TTAA because they're complements. You can see the same thing on the other end. And as a result, this target gene, or this promoter in our case, when it interacts with this plasmid, is going to neatly slide into this gap that we've just created in the plasmid. We use an enzyme called ligase, and it stitches the entire plasmid together. If we do the same thing with the reporter protein, with the reporter gene, we can put that downstream of the promoter, and you can see this is what this will actually look like. This is a nematode called Caenorhabditis elegans. It's one that I studied during my graduate work. What's neat is it's transparent, and what you can see here is they've taken GFP, green fluorescent protein. They've created a transcriptional fusion by fusing it with a promoter that is active during the developmental process, and everywhere you're seeing that bright fluorescent green, that is where that particular gene is expressed. That'll tell you which cell types and when during the developmental process this gene is being expressed. Now, the one thing we have to understand about this is that what you're seeing is a relatively diffuse expression pattern. You can see that it's lighting up the entire cell, and here's why. We have not actually encoded the protein of interest. We've only encoded the promoter for that gene. And as a result, that promoter is going to drive the expression of green fluorescent protein only. Well, green fluorescent protein is not an endogenous protein. It doesn't belong in these cells. So it's just going to spread out all over those cells, just everywhere, like any cytoplasmic protein would. So when you have a transcriptional fusion, transcriptional fusions are great for telling you when and in which cells a given gene is being expressed. What it can't do is create a functional protein. What it can't do is tell you where the actual protein is located at a subcellular level, because it's just the promoter. It's not the actual protein itself. We're just driving expression of an artificial protein called GFP. If we want to know where the protein that this promoter drives is actually expressed within the cell, then we need to create a translational fusion. And translational fusions are a little bit tricky. So quite often we start creating a translational fusion in the way similar to the way that we do the promoter. But instead of just amplifying the gene promoter via PCR, we are going to amplify the gene promoter and the gene of interest. So if we were interested in understanding where protein A is expressed, or when protein A is expressed, we would just take the promoter for gene A and fuse it to GFP. But if we actually want to know where protein A within the cell is expressed, then we need to take the gene promoter and the coding region, the actual gene for protein A, and then fuse both of those to GFP. This is what's called a translational fusion. So again, very, very similar. We identify the promoter region and gene of interest. We examine the plasmid sequence. We need to make sure if we're using 2 specific restriction enzymes or however many we're using, that sites for that restriction enzyme are not within this transgene that we're creating. Otherwise, not only will it cut at the ends, it will cut in the middle as well and destroy the gene of interest. Same thing. I'm not going to repeat this, but the only thing that we need to make sure of is this. We are amplifying the gene promoter and the gene of interest, but we're also going to fuse GFP to the end of this protein. So essentially what we're going to create is the protein of interest, but we also need to make sure that the gene for creating GFP is fused in frame with that gene. Because when it gets translated by the ribosome, we wanted to make the protein we're interested in and make sure that there's a GFP protein attached to the end of it. So to do this, we actually have to mutate the stop codon for our gene of interest so that when it gets to the second to last amino acid, instead of the next codon being a stop, it needs to be the first codon for making GFP. I know this sounds weird, but if you think about it, genes don't know or cells don't know the difference. When they translate, they see the start codon and they keep translating until they encounter a stop codon. So if we mutate the stop codon for our gene of interest and then just act like the first amino acid for GFP is just the next amino acid in our protein of interest, well, guess what? The ribosome is going to translate your protein with a GFP dangling off the end of it, which is a really neat genetic trick that we use when we molecularly clone. And as a result, here's your gene of interest. For example, in this particular study, they're using a gene called Lin39. This is the open reading frame. This is the gene that encodes the protein Lin39. They break the stop codon for Lin39. They just don't include it. And then the next codon is the first codon for GFP. And then it goes all the way until we get to the stop codon for GFP. And as a result, we've now created a translational fusion. This should say translational up here, not transcriptional. Translational fusion. And as a result, this protein is now essentially Lin39 and GFP. And what you can see down here is not only do we know when and in what cells this particular Lin39 gene is expressed in, we can actually see where it's located within the cell at the subcellular level. And what's neat is sometimes if you want to know, you can express these in cells and then use reporters for other parts of the cell. So for example, you might express this GFP, but also label the mitochondria red. So you can see if you have areas where the green and the red overlap, and that would say, hey, Lin39 is expressed in the mitochondria. Or you might use a cytoplasmic indicator to show, oh, Lin39 is expressed in the cytoplasm. Or you might label a different protein altogether with a different color and say, hey, do the green and the red ever overlap, or the green and the blue, or the green and the yellow, do they ever overlap? And that might indicate that those two proteins are actually interacting with each other. This is a really neat way to do a type of study where you look at co-localization of proteins. But this is a trick that's been employed for decades now that allow us to see when and where a particular protein gets expressed. And that might tell us more about how it functions or when it functions or how it does its normal job. Once we've created these transcriptional or translational fusions, before we can put them into our organism of interest or our cell of interest, we first must take that plasmid and amplify it by expressing it in E. coli. This is done through a process called bacterial transformation. So basically, at this point, what we've done is we've created our artificial gene, we've cloned it into a plasmid, and now what we're going to do is put it into E. coli so that we can allow E. coli to make trillions of copies of it. So we'll have lots and lots of that DNA that we can then use to express in our cells or organism of interest. We put the plasmids into E. coli through a process known as transformation. We create competent cells. Competent cells are ones that will allow transformation to occur. So in terms of bacteriology, transformation is a form of horizontal gene transfer in which free DNA can be picked up by E. coli. And the way we typically get the actual plasmid in through transformation is one of two ways. One is by using chemical transformation. This is typically done using calcium chloride. It turns out that when you put cells into high amounts of salt, this could act as a stress that allows transformation. The other one is to allow electroporation. Electroporation essentially zaps the cells with electricity. It reverses the cell polarity, allows them to take up DNA, then locks it in by switching it back. It's a neat little trick called electroporation. So both of these are how they're used. Remember that our plasmids typically contain some type of antibiotic resistance cassette. So for example, we could do the transformation if our plasmid confers some type of resistance to, say, ampicillin, a drug that most E. coli are not resistant to. Most strains of E. coli are not resistant to. You now grow it on media containing ampicillin. It will kill all the bacteria that haven't been transformed. It will only allow the growth of bacteria that contain your plasmid and its ampicillin resistance cassette. And now what you can do is grab those colonies that contain your plasmid, enrich them by growing them in large quantities. So you might grow them in like 500 or 1,000 milliliters of broth to get trillions and trillions of bacterial cells. And then you can isolate them by doing something called a plasmid prep. You essentially destroy the bacterial pellet, you lyse them using detergents, neutralize it, and then isolate all the DNA. And you'll actually end up with this giant glob of DNA at the bottom that you can use to put into your cells or organism of interest. So how do we express transgenes? Well, the answer is kind of simple. The answer is it kind of depends. It depends on what you're trying to do. So if you are working in, for example, mammalian cell culture, you might be looking at a petri dish full of cells, and you can do something called transfection, which is essentially when you grow cells, you grow them in some type of liquid media. You put your plasmid in there, and then you use some type of transfection reagent that will allow the cells to take it up. If you're doing it in nematodes like C. elegans, we microinject. So you can see here, this is a microscopic needle. It is tiny, 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 considering that the distance from this side of the cell to this side of the cell is less than a nanometer. You inject it into their gonad, and it will be taken up into the newly developing oocytes and eventually the eggs, and it will show up in the next generation. In Drosophila melanogaster, which are fruit flies, we typically do this by microinjecting directly into embryos. So the embryos, you take them and then they will hatch. For mice and rats, we typically microinject into zygotes and then implant those zygotes into the mother. And then when the mother gives birth, that transgene will hopefully show up in the next generation. Then what we try to do is establish cell lines or lines for our given species of interest that carry this transgene, and then we use cells or the organism as a whole to be able to investigate things like gene expression, protein localization, and so on and so forth, okay? I am giving a very broad overview of this process, but a lot of people wonder, how do we actually make artificial genes and how do we get them into the organisms? This is how. Now, one of the emerging technologies in the world of molecular cloning and all that is CRISPR. So the CRISPR-Cas9 system is an emerging technology that allows for the highly specific editing of genes. So the one thing that I want you to understand about what I have explained prior to this is that in many cases, these artificial genes that we're putting in are not carried as part of an organism's genome. They often They may not necessarily integrate into the genome. They might be carried as arrays, extra genes, which means they can get lost after a few generations. They don't necessarily show up in generation after generation. They can get lost. So that means you have to re-microinject, re-implant embryos, which is a pain in the butt, right? That's time consuming. It takes a long time to do that. So what we've tried to do is create integrated transgenes. We want them to insert themselves into the host's genome. The problem we have with that is a lot of the ways we do integration is mutagenic. We can't guarantee where in the genome it's going to go, which means it might land inside of a gene that's important for something else. It might land in a gene that might cause a disease because you've disrupted the gene. Really, it's a crapshoot and kind of a pain in the butt. CRISPR-Cas9 changed all that because the CRISPR-Cas9 is technology that was discovered in bacteria. And what's interesting is it actually allows for not only the highly specific editing of genes, but it also allows for the highly specific integration of transgenes. Basically, you can pick the spot you want these transgenes to be added. So what that means is you can create a mutant version of a gene and literally replace the wild type normal version of that gene in a fully functioning living individual, which is a game changer when it comes to being able to create, modify, and insert transgenes into living things. And there are a ton of potential research and medical applications for these. Just go look up CRISPR-Cas9 and go look at the wonderful things that it has the potential to be able to do to treat diseases. There's also some terrifying things that are bioethically questionable that this might lead to. So there's a big interesting debate about that. I'm going to skip a lot of that and just kind of talk about how CRISPR-Cas9 has sort of changed the game for us, help you understand how it works, and then we'll talk a little bit about some potential applications that might be in the near future. So CRISPR-Cas9, in order to use this system, there are three main components. First, you need the Cas9 protein itself. This is an enzyme that cleaves DNA at specific sites. Those specific sites are determined by something called a guide RNA. So this guide RNA can act as a scaffold to allow Cas9 to bind, and then guides the Cas9 enzyme to spots within the genome that you'd like to edit. And then if you're trying to modify a gene, you also want to include template DNA that will allow that to happen. So let's break down these parts a little bit. First, let's start with the guide RNA. So the guide RNA has two parts. It is one large RNA. Part of it is the scaffold. The scaffold is where Cas9 will build, or where it will where Cas9 will bind. This will also help to convert Cas9 into its active conformation, allowing it to bind to DNA. You also need a spacer. This spacer is about 20 nucleotides long. This spacer needs to match the target gene sequence. So creating the spacer is huge because the spacer is going to dictate the specificity of your DNA interaction. So when you build your spacer, you need it to not only match your target, but you also need to make it unique. Because if there is sequence similarity to multiple spots within the genome, that spacer will allow Cas9 to bind to multiple spots within the genome. And if you're trying to specifically edit one spot, that can be problematic because you can get off-target effects, okay? When the scaffold interacts with the Cas9 protein, it causes a conformational change that allows it to bind to the target DNA. And you can see right here, this spacer is now interacting with that target within the genome. And what's going to happen is the Cas9 protein right upstream of where that spacer binds, it's going to create a double-stranded break. So it's going to cut straight through the double-stranded DNA and break the DNA. Now, if you recall from some of my past videos, cells have different ways of handling double-stranded breaks. And what ends up happening is when this double-stranded break gets created, DNA repair mechanisms come into play. The first is something called non-homologous end joining. Now, non-homologous end joining basically, in this, the cells basically go, oh, it's broken, stick it back together. That makes the non-homologous end joining significantly more efficient than other alternatives, but it's also mutagenic because it's not using any sort of template to figure out whether that DNA strand is right. It's just simply saying, I don't know, it's broken, put it back together. So that can be helpful for you if what you're trying to do is disrupt the gene. If you're trying to break it, try to put an insertion or a deletion in there. Non-homologous end joining is very good at producing those indels. But if you are trying to specifically edit a gene, i.e. modify the target so that there's a mutation in there or insert something, then you don't want non-homologous end joining. Instead, what you want is HDR, homology-directed repair. And in HDR, what you're looking at is you are going to also include a DNA template. And with HDR, when it goes to repair that double-stranded break, it is going to go, oh, well, I've got this other piece of DNA here that's going to tell me what this strand should look like. I'll use that as a guide. And then what ends up happening is you can repair that template, but you can kind of trick it. If you want to put a new mutation in there, or you want to put something to correct a mutation, as long as that correction or what you want to be inserted or what you want the gene to say is included in that spacer, you will also, or I'm sorry, in that template DNA, that will get incorporated with that repair process. So if you want to insert a transgene, you can put that in there. And as long as there is homology to the break on either end, so for example, you would create an artificial gene where there's homology here and homology here, but when it goes to repair, you've created this whole big insertion of a transcriptional and translational fusion, it'll set it right in there. Or if you want to mutate the gene so that it encodes a mutation that's similar to something that causes a disease in a human, or if we're dealing with human cells and you want to repair a mutation, you can do that this way. So HDR is best if you're looking to create a targeted mutation, repair a mutation, insert a transgene. The thing we have to watch out for is this. Even when we induce HDR at its highest levels, only about 10% of the cells will actually be repaired through HDR. The other 90% are probably going to get repaired through non-homologous end joining, which could be mutagenic. So it's very, very important if you're using CRISPR to introduce transgenes, to modify genes, to repair genes, that you're screening your population and making sure that the ones that you're investigating or the ones that you're using are ones that were created through HDR and not through NEHJ. There are ways to do this. I'm not going to talk about it here. It's just going to bog down this conversation. But understand which pathway you want to repair depends on what you're trying to do. If you're looking to mutate, want to introduce insertions or deletions, NEHJ is your way to go. Non-homologous end joining is great. But if you're trying to specifically edit a gene to say something different or say what you want it to say, you need it to be done through HDR. Non-homologous end joining will not do that for you. CRISPR can also be used to label and visualize chromatin or to visualize RNA dynamics. This is another potential application. To do this, they use something called dCas9. So what's interesting about this is dCas9 is catalytically inactive. It cannot cut the DNA. But if you provide a guide RNA, it will still bind to the target sequence. It just won't do anything about it. But if you take if you take dCas9 and you fuse GFP to it, it will bind to the target DNA so that you can know when this gene is being expressed. Because you can put fluorophores either on the scaffold, you can put fluorophores directly on dCas9, and in doing so, it'll say, okay, this is when this particular gene is being interacted with. This is where it's expressed or when it's expressed, which is kind of neat. You can also use dCas9 to introduce chromatin modifications. So let's say you want to see what happens if you deacetylate, methylate, acetylate, do all these different things. You take dCas9, you modify it by attaching either an HDAC or a histomethyltransferase or an acetyltransferase to that dCas9. Then when the dCas9 interacts with that specific promoter region that you've labeled with your guide RNA, it will then acetylate or decetylate, methylate or demethylate. And then what you can do is go, okay, well, what happens if we demethylate this particular promoter region, we get this expression pattern, which is also really, really neat. So what I'm showing here is how do we get CRISPR into cells? Well, again, that also depends. So if we're talking about this strictly from a lab standpoint, we can use things like electroporation, liposuction, microinjection, things like that. But we are now getting to the point where CRISPR might be able to be used therapeutically. And we are now beginning to experiment with what happens when we try to induce CRISPR-generated transgenes into living organisms. And this can be done through nanoparticles, or it can be done through the use of genetically modified viruses. And these are the two major platforms we're looking to right now to see if there are ways to actually use CRISPR as a way to edit the genomes of living organisms, including humans. So what I want to end this conversation with is, what are some potential applications for CRISPR in human beings? What are some things that CRISPR might be able to do? Well, here's a great example of this. All right. CRISPR gene editing has many potential therapeutic applications. One of the ones that has a lot of high potential for being used in the near future is for patients who have inherited red blood cell diseases. All right. There are lots of different inherited conditions, such as sickle cell anemia, beta thalassemia. These are red blood cell diseases. And currently, the way that these are being treated genetically is through something called gene therapy. And essentially what they're doing is they're using gene therapy to correct hematopoietic stem cells, which red blood cells and all other blood cells come from, using lentiviral vectors. All right. Now, this allows these modified cells are then reintroduced into the host where they can overcome the deficiency caused by that particular mutation. Okay, so we basically create healthy red blood cells, or at least red blood cell progenitor cells, put them back into the person. And then for a period of time, these healthy versions can create healthy red blood cells that sort of outweigh the mutated population of red blood cells. Now, there are some problems with this. First and foremost, there's no ability to integrate this transgene, which means these hematopoietic stem cells are around for a while, but eventually they'll go away and the process needs to be repeated. It also doesn't allow for endogenous regulation of the gene. Essentially, what you're doing is you're putting extra healthy copies of a broken gene into an individual, and we can't really regulate how much or how little this is expressed. So as a result, this type of therapy is only moderately effective because we really can't control things like dosage or for how long these healthy cells are going to be around. CRISPR can change all of that. And here's how this might look. The CRISPR-Cas9 system will allow for targeted modification of hematopoietic progenitor stem cells. These are the cells that hematopoietic stem cells come from. Okay, and these ones have the ability to accumulate and reproduce within a patient's bone marrow. So if we take out these HSPCs from an affected individual, we isolate them, we use CRISPR-Cas9 mediated gene therapy, and essentially in these cells, we replace the broken version of the gene, the mutated version, and replace it with the wild type version of this gene, and then reinfuse these HSPCs into a healthy individual. Now what's going to happen is that generation after generation of these hematopoietic stem cells will come out of this, and they will steadily replace the mutated version in that person's bone marrow. Essentially, you're introducing a fix for life. Here's why this is also cool. This is being done in the patient's cells ex vivo, which means we can integrate this into the red blood cells or into these HSPCs, but the rest of the person's body is unaffected. We're doing this outside of the body, doing the integration into these HSPCs and then putting the cells back in. If we were to do this in the person's body, all of the cells could be subject to this type of correction. That could theoretically be problematic. But because we're doing it ex vivo, we're doing it outside the person, we can do it in cells, make sure the cells look right before implanting them, then put them back. And essentially, this person's body is set for life. This fixes the problem with these red blood cells, but doesn't introduce any additional risk to mutating the other patient's somatic cells. And in this way, we might be able to correct inherited red blood cell diseases like beta thalassemia, like sickle cell, permanently in this population. Now, does this introduce a whole bunch of other bioethical concerns? It does. Because off the bat, this type of technology will not be cheap. So questions start to arise. Should this be something that's available for everyone? And if so, who's paying for it? If it is something that's only available to wealthy people that suffer from these, what about people all over the world that can't afford this treatment? Is it morally ethical for us to allow people to suffer and in some cases die from diseases that we know we could treat if they just had more money? So there's a lot that goes into these and into this conversation that I'm not talking about in terms of bioethics and things like that, but these are questions that we're going to have to confront as a society as these particular medical interventions become more and more possible. So anyways, I hope this video gave you a good idea of like, hey, I see like green fluorescent cats and plants that grow in the dark. How do we create these transgenes? And to be clear, those are novelties. Molecular cloning is done every single day in laboratories throughout the world in order to help us better understand the expression pattern of genes, the function of certain proteins, and understand how these things are regulated within a living cell or within a living organism. CRISPR has now made it possible for us to possibly create artificial genes that would allow us to repair genetically encoded diseases and possibly prevent the lifelong suffering of millions of people. We're still working on this technology. There's essentially limitless applications for CRISPR, both good and bad. But it all goes back to our initial understanding of how molecular cloning worked. And a lot of the technology that goes into creating these CRISPR transgenes goes right back to being able to have a fully sequenced genome, using PCR to amplify that, and creating artificial guide RNAs, and so on and so forth. I hope you enjoyed this conversation. I hope you found it educational. And I hope to see you watching other videos of mine. Thanks for watching, and I'll talk to you again soon.